Hello students, this is Mr. Dohmen. This is your first lecture for this week. We are going to be discussing DNA and RNA structure. Later this week we'll be looking at DNA replication. But right now our focus is to get you guys to identify the differences between DNA, RNA, understand the structural components and make them both up, and give you a little bit of background information on uh, both types of genetic material. So what is DNA and RNA? Well, there are two kinds of genetic material, DNA and RNA. DNA stands for deoxyribose nucleic acid. It makes up the genes, which makes up chromatin, and is packaged into chromosomes. Okay, so the hierarchy is that you've got your individual uh, segments of DNA. Those segments make up genes. Those genes make up chromatin, and the chromatin itself is what DNA looks like when it is not being replicated for mitosis. But once it condenses, when the chromatin condenses, that's when it gets packaged into chromosomes. RNA stands for ribose nucleic acid. RNA is used by cells for certain processes and is the genetic material for many viruses, as you learned last week. So we're first going to do a little bit of history about DNA, specifically when it was first discovered. Scientists were trying to understand how heredity worked in living organisms. In other words, how were traits passed from parent to offspring? Okay, which is what heredity is. So scientists first identified chromosomes as the source of the traits being passed from parent to child, and eventually figured out that they were made up of DNA and proteins. Yet no one was able to really identify what DNA looked like. In the 1950s, however, a scientist by the name of Rosalind Franklin, she came up with the first photograph of DNA using a process called X-ray crystallography. Okay, she had taken many photos, but the one that clearly identified what DNA looked like was called photograph 51. Um, Shortly thereafter, about 2 or 3 years later, two other scientists, Watson and Crick, using um, Franklin's photograph 51, they were able to develop the first 3D model of DNA. So looking at the structure of DNA and RNA, well, DNA, uh, it has a deoxyribose sugar. It has a double helix shape. The four bases that comprise DNA are adenine, guanine, thymine, and cytosine. Um, and we represent these with the letters A for adenine, T for thymine, G for guanine, and C for cytosine. Okay. Between these bases, we have weak hydrogen bonds that allow for the bases to be connected and thus connecting the two strands of DNA. The strands run anti-parallel to each other. What do we mean by anti-parallel? Basically, what it means is the two strands run in opposite directions of each other, even though they essentially are parallel. One strand runs in a direction of 5' to 3', and that's what those apostrophes here mean, folks, as prime. And then the complementary strand runs in the opposite direction of 3' to 5'. Okay, and we'll look at that here shortly when we look at the images of DNA. The helices can also twist in a right-handed or counterclockwise direction or a left-handed or clockwise direction. As a result, we have three different types of DNA based on that handedness. We have aDNA, bDNA, and zDNA. This is not going to be crucial information, just something I thought you might find interesting. Okay. RNA, on the other hand, is composed of ribose sugar. It is single-stranded. You would notice somewhat helical, and unlike DNA, which has thymine, RNA uses uracil instead, okay, which we represent with the letter U. So if we take a look at the two strands of genetic material side by side, here on the left we have RNA, and on the right side here we have DNA. Okay, these thick, dark-colored strands that we see here, these are the backbones of DNA, which consists of the 5-carbon sugar and a phosphate group. Okay, so on DNA, this is where the deoxyribose sugar is found along with phosphate. And on RNA, this is where the ribose sugar is found with a phosphate. Okay, and those two parts make up nucleotide, which we'll take a look at shortly. Okay, as you can see, RNA is single-stranded yet still twisted in a helical shape. DNA, double-stranded, twisted in a helical shape. Okay. Now, earlier I mentioned that the strands run 5' to 3'. Okay, so looking at this, we could say that this image here shows a right-handed DNA strand, which means that this end here is our 5' end. This here is the 3' end of the complementary strand. So if we follow this strand all the way up, that would make this end here the 3' end, and this would be the 5' end of the complementary strand. Okay, so as you can see, they run anti-parallel to each other. All right, now here are the structures of the nucleotide bases. As you can see, cytosine, guanine, and adenine are both shared by RNA and DNA. Thymine and uracil are different. Now, what I want you to notice, the biggest difference between these two bases is this one side chain here. Here we have a methyl group that's on thymine, but it's missing on uracil. Otherwise, they're both identical in every other way. Okay, so that is why going from DNA to RNA, we can pair adenine with uracil, which we'll talk about here in a second. Okay, so we've been talking about base pairs, right? Well, those base pairs, we refer to them as nucleotides. Okay, and they're made up of 3 components. There's a phosphate group, which is what this is right here. Okay, this is a phosphate group. Well, actually, I should include that oxygen in there as well. Then we have our 5-carbon sugar, which is this part right here. Okay. And as you can see in this case, this is the deoxyribose sugar in this case. Well, for the bases, it would be a deoxyadenosine triphosphate. Okay, so remember that molecule we talked about, the energy molecule ATP, right? This is basically what that is here. It's just a modified version of ATP. Okay, and then over here is our nitrogenous base. Okay, now if we look in this example here, well, this base is clearly adenine because this structure and this structure match. Okay, the phosphate group and the 5-carbon sugar group, they make up the backbone of both DNA and RNA. Okay, these bases here, on the other hand, run as ladder rungs for both DNA and RNA. Okay, and there's 5 bases total again: adenine, guanine, cytosine, thymine, and uracil. They're also divided into 2 subgroups: purines and pyrimidines. Okay, cytosine, thymine, and uracil are all pyrimidines because they have a single ring structure. Okay, single ring structure. Purines, on the other hand, have a 2-ring structure. Okay, so when we say rings, we're talking about these carbons here and nitrogens that are in a somewhat circular shape. Okay, so you can see this ring here has 6 sides, so it's a hexagon. This ring here has 5 sides, it's a pentagon. And then this ring here is a hexagon. Okay, so adenine and guanine are your purines, while cytosine, thymine, and uracil are your um, pyrimidines. Okay, adenine will pair with thymine and uracil, whereas guanine and cytosine pair together. We'll look at that in just a second. And of course, here are your sugars, your ribose, which is found in RNA, and your deoxyribose, which is found in DNA. And as you can see, they're pretty much identical with the exception of this hydroxyl group and this hydride here. That's how we are able to tell the difference between the two sugars. Otherwise, they're identical in every other way. Okay? So the bases that we just looked at, they pair up in what we call complementary base pairing. And that's misspelled. That should be an E. Sorry, folks. Bases pair according to their structure. Adenine always pairs with thymine and DNA, and then with uracil and RNA. And then guanine always pairs with cytosine regardless of the structure, be it DNA or RNA. Okay. And we have a mnemonic device that will help you remember this. To remember that adenine pairs with thymine, we always say apples in a tree, or A to T. And to help you remember that adenine pairs with uracil, we always say apples under the tree, or A to U. Okay. And then lastly, guanine pairing with cytosine, we always say cars in a garage, because you know that's where you park a car, is in a garage. All right. So you can see here how they pair up. Guanine pairs up with cytosine because you've got 3 weak hydrogen bonds here. They pair with these side groups. And then thymine and adenine pair up with 2 hydrogen bonds between these 2 side groups. Okay. Now we have something called Chargaff's rules, which explains the mathematical composition of DNA based on the nucleotides. It was originally thought that the amount of adenine, thymine, cytosine, and guanine were in 4 equal parts. The Austrian scientist Chargaff realized that this was not true, that they were not found in equal quantities. In fact, the amounts of the bases varied among species, but were not different, or that much different between individuals of the same species. What does this mean? In other words, it means if we were to compare the amount of adenine, thymine, cytosine, and guanine among all human beings, we would see that roughly all of us have the same amount of adenine, thymine, cytosine, and guanine. But if we were to look at, say, the genome of like a dog, for example, we would see that they have a completely different percentage of adenine, thymine, cytosine, and guanine compared to humans. All right. But Chargaff did find that the amount of adenine was always equal to the amount of thymine, and that the amount of cytosine was always equal to the amount of guanine. Okay, so A equals the amount of A equals the amount of T, and the amount of G equals the amount of C. Okay. So anytime that you come across a problem asking you, if you've got 20% adenine and 30% guanine, then what are your totals going to be of cytosine and thymine? Then would be 20 and 30% respectively. Okay. That pretty much covers DNA and RNA structure. I want you to go back to Canvas and click on the next button in the lower right corner and look over your assignment for DNA structure. You guys take care.